Okay, so now we will be discussing oxidative deamination. Okay, what is oxidative deamination? It's a process. Oxidative deamination is a process by which the amine groups of amino acids are converted into ammonia, which are water soluble. Clear? Now listen. After oxidative deamination, what we will learn is how this ammonia is converted into urea, so that it can be excreted through your kidneys. Clear? Now listen. Whenever you are trying to understand oxidative deamination, you must understand why we call it oxidative deamination. Now listen. Actually, as there is oxidative deamination occurring, there is reductive deamination as well, but we will talk about that later. Okay. Now, for the time being, what we will learn is how this oxidative deamination actually occurs. Okay. How this oxidative deamination actually occurs, this is our most important topic right now. Now listen. Now oxidative deamination actually takes place. Now listen, there are two sites available for oxidative deamination. Site 1 is the organs where oxidative deamination takes place, and the organs are liver and kidney. Clear? Now the site 1 is organ, which is liver and kidney. Now the site 2 is actually the molecular site. Okay, the site 2 is actually the molecular site. Okay, molecular site, that is mitochondria. It occurs in mitochondria. Clear? That means oxidative deamination takes place in liver and kidney in the organ level, and in a cell oxidative deamination takes place in mitochondria. Clear? Okay. Now we will be talking about in details the oxidative deamination process. Okay. Now the first thing we need to understand is this: oxidative deamination starts from a standpoint which is the glutamate. Clear? That means what we must understand right now is this, that glutamate is the starting point of oxidative deamination. That means glutamate will lose the amine group and will form something. Now what is that something? That something is nothing but a keto acid. Clear? Again, now listen with all your attention. That means glutamate is now converted into keto acid. Let me draw it. Okay. Now in case of oxidative deamination, what you will have to learn is that in case of oxidative deamination, the glutamate is converted into a keto acid. Okay, the glutamate is converted into a keto acid, but what we have seen is in other, in transamination, what you have seen is a keto acid is being converted into an amino acid, right? This was your transamination process, right? But in oxidative deamination, this doesn't happen. In oxidative deamination, what happens is when you want to convert an amino acid into keto acid, then in oxidative deamination, you don't need any keto acid. What you need is an enzyme which can just simply take out or kick off the amine group present in the amino acid. That's it. It will have to just kick out the amine group. Okay. Now the enzyme required for this particular process is known by the name of glutamate dehydrogenase. Okay, glutamate dehydrogenase. Now a very important perspective you need to understand right now is this, that glutamate dehydrogenase is a very, very peculiar type of enzyme. Okay, it's a very, very peculiar type of enzyme. It's a very, very peculiar type of enzyme. Why I am saying this and why I am stressing on this? Because the cofactors associated with this, glutamate dehydrogenase helps in oxidative deamination as well as reductive deamination. We'll talk about these in details now. Now listen. So we will now see what is the role of glutamate dehydrogenase. What is the role of glutamate dehydrogenase in oxidative deamination? Okay. We will be talking about role of glutamate dehydrogenase in oxidative deamination. Okay, oxidative deamination. Okay, now the first things first, that is we will be talking about glutamate dehydrogenase role in oxidative deamination. Now first thing is this: if we don't learn how to draw glutamate, then we cannot understand why it is called oxidative deamination. Now according to organic chemistry, oxidation means what? Removal of hydrogen or addition of oxygen. According to organic chemistry, what is oxidation? Oxidation is removal of hydrogen, right, or addition of oxygen. This is your standard definition of oxidation, right? Right, now listen. Which of the two definition will fit in this category? That is, which one of the two definitions, that is removal of hydrogen or addition of hydrogen or addition of oxygen, will occur during oxidative deamination? Now listen. During oxidative deamination, both will occur. At the same molecule, hydrogen will be released and oxygen will be introduced. Clear? Now how? Suppose this is your glutamate, okay? Glutamate is, we have already learned, CH2, CH, COO minus, NH3 plus, okay? Now listen with all your attention. A very important point. So this is glutamate, an acidic amino acid, right? If we remove this CH2 group, then it will become aspartate, right? Now, suppose this is your mitochondrial, matrix. Suppose we want to convert this into a keto acid. What we will have to do? Again, simple thing. We will have to take out this particular, group and introduce a keto group here. That's it. We'll have to introduce a keto group here, right? Now, a very important point you need to understand here is this, that if you draw the structure like this, that is, this is your CH2, this is your CH2, this is COO minus H, sorry, this is your, keto group, okay? So this is your, structure. Now what is this? What is this? This is nothing but alpha ketoglutarate. This is nothing but alpha, this is alpha carbon, alpha ketoglutarate. Why it is alpha ketoglutarate? Just count the number of carbons, 1, 2, 3, 4, 5. Oh my son, go and play. Simply apply oh my son, go and play. Ohm's gap. Okay, that is, let me tell you, oh my son, go and play. O for, it's a homologous series. We have already discussed in details what is Ohm's gap in your fatty acid synthesis video. Now listen. Oxalic acid, this is dicarboxylic acid homologous series. M is just introduced one CH2 here, Again introduce another CH2 here. Now glutarate or glutaric acid. Introduce how many CH2? This is 1 CH2, this is 2 CH2, right? This is 2 CH2, sorry, I have accidentally drawn the glutarate already, okay? And this is your glutarate, right? Or glutaric acid, sorry. Okay, so this is your glutaric acid. Now important point you need to understand is this: if you want to convert this glutaric acid into alpha ketoglutarate, what is this? This is primary functional group, or this is the only functional group, and just the carbon just beside the primary functional group or functional group is alpha carbon. So there are 2 alpha carbons. Now if you convert one of the alpha carbons into a keto group, then it is alpha ketoglutarate, alpha Kg. Okay, this is alpha ketoglutarate, right? Now a very important point here you need to understand is this, that so this is your alpha ketoglutarate, right? Now alpha ketoglutarate is what? A keto acid. A keto acid, right? It's a keto acid. It's a keto acid. Why? It is an acid containing keto group. Now why it is called a keto acid? Because the acid is the primary functional group here. Now listen, these acid groups are the primary functional groups, okay? Primary functional group, and this keto group here is the secondary functional group, That's why whenever we write, we always write alpha keto acid, okay? It is not looking like alpha. Okay. It is called alpha keto acid. Why? Because the secondary functional group comes first, then the primary functional group name comes, okay? That's why we don't talk about alpha carboxylate, ketone. We talk about alpha keto acid. Why? Ketone group, or not the ketone, actually, it is keto group. This keto group is actually a secondary functional group here. Clear? Now, the enzyme involved in the conversion of this glutamate into alpha ketoglutarate is known by the name of glutamate dehydrogenase, which is present in your mitochondrial cytosol, right? Or mitochondrial matrix. Now, the first point you need to understand is this, that glutamate dehydrogenase is the enzyme which is essential for the conversion of what? For the conversion of glutamate into alpha ketoglutarate, right? Now a very important point here you need to understand is this glutamate dehydrogenase enzyme also has a very important coenzyme, which is NADP. Now listen with all your attention. The cofactor of glutamate dehydrogenase is actually NADP plus, and also there is another cofactor, that is NADPH. Confused, right? That means glutamate dehydrogenase is such an enzyme, it is laughing. Okay, now listen, glutamate dehydrogenase is such an enzyme which has two coenzymes. This enzyme is very much unique because it has got 2 coenzymes. It has got 2 coenzymes, not one. Why? Now listen with all your attention. One thing I told you at the beginning of this lecture, that is this glutamate dehydrogenase performs 2 things, that is oxidative deamination as well as reductive deamination. It performs 2 things, that is it actually helps in oxidative deamination as well as reductive deamination. Now listen, when it is suppose I am glutamate dehydrogenase, okay? Suppose I am glutamate dehydrogenase, okay? Now if I am performing oxidative deamination, that means I will be performing oxidative reaction or I will be performing an oxidation reduction. Now If I perform reductive deamination, I will show that. If I perform reductive deamination, then I will be performing a reduction reaction simply. That means what I am trying to say is this, that suppose I am glutamate dehydrogenase and I am performing, oxidation as well as reduction at the same time. That means I am performing a redox reaction. I am performing a redox reaction, right? Redox means reduction and oxidation at the same time, okay? Redox reaction. Now, important point here you need to understand is this, that whenever glutamate dehydrogenase perform oxidative deamination, whenever glutamate dehydrogenase perform oxidative deamination, the first thing you need to understand is this, it is actually taking out hydrogen. It is actually taking out hydrogen and introducing oxygen, right? That means whenever glutamate dehydrogenase is performing oxidative deamination, it is performing oxidative deamination, then what is happening? Then it is taking out oxygen. It is taking out oxygen. Sorry, it is taking out hydrogen and introducing oxygen, right? It is taking out hydrogen and introducing oxygen. That means there should be someone to take that hydrogen out of this vicinity. There should be someone to take this hydrogen or H plus out of this, whole scenario. That means when glutamate dehydrogenase is performing oxidative deamination, then which coenzyme will work, NADP plus or NADPH? Now listen, it's a very easy, answer. Which has got H already? NADPH has already got H. That means it will no longer be further loaded with H plus. Now this requires H plus. That means whenever glutamate dehydrogenase is performing oxidative deamination, you must understand a simple thing, that is whenever, now listen with all your attention, whenever glutamate dehydrogenase perform oxidative deamination, the first thing that should come into your mind is this coenzyme will work. There are two coenzymes. Now this enzyme, suppose I am glutamate dehydrogenase, I have two hands. Now I can use both these hands, but depending on the scenario, I can use one of my hands. Okay, suppose I will be performing oxidative deamination. Suppose in this hand I am having NADP plus. That means I will be now using this hand instead of this. Clear? So here NADP plus will be converted into NADPH, and this NADPH can go on to give you ATP, right? Now we'll talk about that, how this thing controls everything. Now, so whenever glutamate dehydrogenase is performing oxidative deamination, then NADP plus, this thing is working, that is this coenzyme is working. Okay, now so this is oxidative deamination. Okay, but how can you discuss, or how can you understand that what is the function of glutamate dehydrogenase when reverse is occurring? Now listen, when glutamate dehydrogenase is performing oxidative deamination, NADP plus is getting converted into NADPH, and another thing is going out. That is NH3 plus. This NH3 plus is going out. Okay, this NH3 plus is going out, and this H is actually going out as, okay, and here a keto group is introduced. Clear? Now, a very interesting part is this. If you want to understand really how glutamate dehydrogenase helps in reductive deamination, suppose if you want to know how glutamate dehydrogenase helps in reductive deamination, then the most important thing you have to understand is this. You will have to do one thing. You will just have to reverse this arrow. That's it. You will just have to reverse this arrow. Clear? That means whenever glutamate dehydrogenase is performing oxidative deamination, ammonia will go out. But whenever it will perform reductive deamination, ammonia will come in. Ammonia will come in, and this keto group will be converted into amine group. Okay. Now important point you need to understand is this, that how this thing actually happens. Now listen, in this scenario, when suppose this is reaction 1, okay, let me draw with another color. Okay, suppose this is reaction 1 and this is reaction number 2. Okay, reaction number 2. Now important point you need to understand is this, suppose this reductive deamination is occurring right now. The first and the most important thing you need to understand is this: during reductive deamination, which coenzyme will work, this or this? Simply this. Why? Because this particular alpha ketoglutarate now needs more hydrogen, and therefore whenever reductive deamination works, NADPH will be converted into NADP plus. Clear? Will be converted into NADP plus. Okay? Clear? Right? Up to this, it is clear? Okay. Now, a very important point here you need to understand is this, that when you are talking about glutamate dehydrogenase, you must understand that this glutamate dehydrogenase is present in the mitochondria, and mitochondria is the hub of producing ATP, right? Mitochondria is the hub of producing ATP. Now listen, a very important point here you need to understand is this, that this enzyme can sense that when a cell needs energy and when a cell is already filled up with more energy, that is, it can sense whenever cell requires more ATP, and it can also sense whenever cell does not require any ATP. Clear? Now listen, what I am talking about is this, the allosteric regulation of glutamate dehydrogenase. Clear? Now we will be discussing the allosteric regulation of glutamate dehydrogenase. Now listen, if you are talking about allosteric regulation of glutamate dehydrogenase, okay, allosteric regulation, suppose you are talking about allosteric regulation of glutamate dehydrogenase, the first thing you need to understand is this, suppose this is your glutamate dehydrogenase. Clear? Suppose this is your glutamate dehydrogenase. Now glutamate dehydrogenase, what it does is, glutamate dehydrogenase converts glutamate into alpha ketoglutarate, and this is oxidative deamination, and in the process it releases NH3 plus, and the reducing equivalents are NADP plus to NADPH. Clear? Now a very important point here you need to understand is this, that glutamate dehydrogenase is an allosteric enzyme. And now listen, now listen with all your attention, whenever glutamate dehydrogenase is converting glutamate into alpha ketoglutarate, that means alpha ketoglutarate is what? An intermediate of TCA cycle. Now when alpha ketoglutarate enters TCA cycle, it produces compound known by the name of GTP, guanosine triphosphate, right? From TCA cycle we get GTP. Now listen, this GTP inhibits glutamate dehydrogenase. That means, now listen, why this thing, or why this type of allosteric inhibition occurs, or why this allosteric inhibition is important. Now listen, whenever glutamate is converted into alpha ketoglutarate, now listen, whenever glutamate is converted into alpha ketoglutarate, the first thing you need to understand is this alpha ketoglutarate can now enter into TCA cycle, and whenever it is undergoing changes in the TCA cycle, in the meantime it can give rise to GTP or guanosine triphosphate. Now listen, presence of GTP is a very good example of, you know, energy rich sources in the cell. Guanosine triphosphate, adenosine triphosphate, all these are what? Nucleotides, right? Now listen, whenever the concentration of GTP increases, then GTP actually inhibits this glutamate dehydrogenase. Clear? GTP inhibits this glutamate dehydrogenase. Okay? Now, that means glutamate dehydrogenase will be inactivated. Now listen with all your attention. Glutamate dehydrogenase will be inactivated when cell has got its, you know, energy levels right. Whenever the cellular energy level of a cell is absolutely correct, then no amino acid will be destroyed and no amino acid will be deaminated, or no amino acid will be deaminated for the carbon skeleton of its, you know, amino acid, so that these alpha ketoglutarate cannot enter TCA cycle. That means whenever the cellular energy levels are right, then amino acids will not be broken down. Into alpha ketoglutarate, and deamination will not occur. Clear? Now, if we are fasting for a long time, then amino acids are broken down to give energy to our system, right? Now listen, how this, you know, inhibition is lost. Now listen with all your attention. Suppose you are fasting for a long time, then what will happen is this. You need to understand is this. Then what will happen? Your cellular energy levels means what? ATP. When you're fasting for a long time, then all your ATPs will be burned out, and the concentration of ATP will be low, and the concentration of ADP will be high. Why? Because this ATP has been converted into ADP, right? This ATP has been converted into ADP, right? Because of low energy, this ATP has been converted into ADPs. Now, important point you need to understand here is this, that when these ATPs are converted into ADPs, then this ADP, this intracellular concentration of ADP will activate glutamate dehydrogenase. That means whenever the cellular energy levels are low, then ADP is more than ATP, and this ADP, you know, activates glutamate dehydrogenase. Okay? And when this glutamate dehydrogenase is activated, then amino acids are converted into alpha ketoglutarate, where they can enter into TCA cycle, and GTP is produced further, and then they can inhibit. Okay? This is how the allosteric regulation of glutamate dehydrogenase actually occurs. Now, a very important point you need to understand is this, that whenever we are talking about amino acids, that is those amino acids which give rise to, you know, alpha ketoglutarate or something like that, there is a difference. Why? Because all these amino acids, whether it is D or L, in most of the cases our body accepts L amino acids. Okay? But suppose this is a plant, okay, or animal, you know, whatever it is, animal part we are eating. Now this particular, this is suppose plant part. Now you know there are you know, protein powders which have plant sources. Now listen with all your attention. The company is fake, you know, who is selling these plant proteins. Why? Because most of the plant proteins contain D amino acids. And we don't have the machinery to use D amino acids. D amino acids never get absorbed properly. First, and the most important part you need to understand is this: D amino acids are converted into alpha ketoglutarate, so that they can enter TCA cycle. That means plant proteins are the worst, you know, one of the worst sources of protein. Why? Because plant proteins will never give you the proper, you know, amino acid balance. Why? Because most of the plant proteins have D amino. They also have L amino acids, no doubt about that, but they also have D amino acids. That is the point. Clear? That is why it is very important for you to understand that whenever you are buying any protein supplement or something like that, you should understand that you should check the constituents. If your protein supplements are saying amino acids like arginine, lysine, then you should check whether they are D-lysine or L-lysine. Your body absorbs L-amino acid. Clear? Your body absorbs L-amino acid. Okay. Now, most of the D-amino acids are converted into this carbon skeleton. Clear? And they enter TCA cycle. Okay. Now, why plant proteins are useful? Can you say why plant proteins are useful? Now listen. From plant protein, if you digest plant protein, you will get amino acids, but they are in most of the cases they are D-amino acids. But there is an important thing. A very intelligent person actually devise these plant proteins. Why? Because plant proteins will be converted into alpha ketoglutarate or any carbon skeleton, right? Will be converted into any carbon skeleton, and this carbon skeleton may enter TCA cycle, and that's why they will inhibit glutamate dehydrogenase, and in turn they will actually inhibit the destruction or the conversion of normal amino acids into alpha ketoglutarate. Therefore, your muscle wasting will stop. Clear? Okay. So now we will be talking about those enzymes which deal with this, D-amino acid, deamination. That is amino acid oxidase. Okay. We are talking about amino acid oxidase.